So picture an animal that has basically figured out how to completely cheat the fundamental rules of life.
Oh, yeah, the green sea slug.
Yes, the green sea slug, Elysia chlorotica. I mean, it crawls along the ocean floor eating algae. But instead of just digesting that algae for, quick burst of calories, its digestive tract selectively filters out the algae's chloroplasts.
Right, which are basically the microscopic solar panels that plants use to make food from sunlight.
Exactly. And the slug just shuttles these completely intact chloroplasts outward into its own skin. And suddenly this animal doesn't even need to eat anymore.
It just sunbathes.
It just sunbathes. It becomes
Yeah.
a solar-powered animal.
Mm.
But here's the truly insane part. Chloroplasts break down, right? Like they need constant maintenance from specific proteins that are coded in the plant's nucleus.
And the slug obviously didn't eat the nucleus.
Right. So how does it maintain the chloroplasts for like up to a year?
Yeah.
It turns out over millions of years, this sea slug physically stole the required maintenance genes from the algae and just pasted them into its own animal DNA.
I mean, it is the ultimate biological heist. It completely shatters the neat vertical lines of inheritance that we're always so used to mapping out.
Yeah.
We call it horizontal gene transfer. And it basically proves that the boundaries between species, the very walls protecting the genetic code, are just, well, a lot more porous than we ever assumed.
And that tension, right, between the strict, rigid rules of biology and the totally chaotic, brilliant ways life just breaks those rules, that is exactly what we are diving into today.
Absolutely.
Because look, if you are listening to this, there is a very good chance you are staring down a massive biology unit test on genetics, heredity, cell division, and DNA.
Oh, the classic unit test.
Yeah, you've probably got a mountain of textbook chapters in front of you, and it feels like you're just, I don't know, memorizing a telephone directory, a disconnected term.
Which is pretty much the absolute worst way to understand biology. I mean, biology isn't a list of vocabulary words. It is an unbroken, billions of years old narrative about information management. So today, we are basically going to act as your study team for this deep dive.
We're going to synthesize all those dense chapters into a single, cohesive story.
Exactly. We are looking at the eternal war between biological perfection and biological chaos. You know, how a cell copies its code flawlessly, why it occasionally just scrambles that code on purpose.
Which is wild to think about.
It really is. And we'll cover how those invisible genes actually translate into the physical traits we see. And finally, what that code actually looks like down at the atomic level.
Perfect. So let's start with the quest for perfection, right? The clone factory. Because before we even touch on how traits mix between parents, we have to look at how an organism simply replicates its own genetic blueprint.
Right. Basic asexual reproduction.
Yeah. And the textbook highlights asexual reproduction, and the aphid is really the ultimate case study here.
Oh, the aphids are terrifying.
They really are. Because in the spring, a female aphid gives birth to live identical female offspring without even mating.
Yeah, and it gets weirder.
Right, because those offsprings are born already pregnant with the next generation of identical females.
Telescoping generations.
Yes, they telescope the generations. They can pump out, 41 generations in a single year?
Easily. It turns one aphid into literally a trillion.
It's just a terrifyingly efficient strategy. I mean, asexual reproduction sacrifices genetic diversity for just sheer numerical supremacy.
Exactly. And to pull that off, the aphid relies on mitosis. Mitosis is basically the engine of perfect replication.
Right.
But to understand the sheer mechanical challenge of mitosis, you have to think about what the DNA actually looks like inside the nucleus during interphase.
Like before the cell even starts dividing.
Right, because it isn't neatly packaged. It's chromatin. It's just a loose, sprawling, microscopic tangle of thread.
Which makes sense, because if the cell is just, living its life, it needs to be able to actually read the genes to make proteins.
Exactly. You can't read a book if it's packed tightly into a moving box.
Right. But when it's time to divide, you can't just pull a messy ball of yarn in half.
No, it would snap. You lose millions of base pairs of data instantly.
So that is the genius of prophase, right? The first stage of mitosis.
Precisely. During prophase, the cell takes that massive, tangled chromatin and hypercoils it around specialized proteins called histones.
Like wrapping a garden hose around a spool.
Yeah, exactly like that. It packs it incredibly densely into the distinct X-shaped chromosomes we all recognize from textbooks.
And we should keep in mind, during interphase, the entire genome was already duplicated.
Right, the replication already happened.
So that X shape is actually two identical copies, right? Sister chromatids. held together at the waist by a protein complex.
The centromere.
The centromere, right. Okay, so the cell has boxed up the library. But how does it physically move these massive, heavy chromosomes around? Because the nucleus dissolves, and these things just seem to magically align in the center of the cell during metaphase.
Well, it isn't magic, it's molecular machinery. So during prophase, structures called centrosomes migrate to the opposite poles of the cell.
Okay.
And they begin extruding these microtubules, which are spindle fibers. Think of them as dynamic, self-assembling steel beams.
Oh, wow.
Yeah, and these fibers grow outward until they physically crash into a specialized protein patch on the centromere.
Which is called the kinetochore, right?
Exactly, the kinetochore.
So they basically harpoon the chromosomes.
Yes, from both sides. And then this microscopic tug of war begins. Motor proteins on the kinetochore literally walk along the spindle fibers.
Wait, they walk?
They do. They pull the chromosome back and forth until the tension from both poles is perfectly equal.
And that equal tension is what forces all the chromosomes to line up perfectly along the equator of the cell during metaphase.
Right. It's just a mechanical equilibrium.
Which sets the stage for the split, anaphase. The central proteins holding the sister chromatids together dissolve. But the spindle fibers don't just act like bungee cords snapping back, do they?
No. It's a combination of mechanisms. The kinetochore motor proteins act kind of like little Pac-Man.
Okay, I like that visual.
Yeah, they chew up the spindle fiber and walk the chromatid toward the pole. Simultaneously, other spindle fibers that aren't attached to chromosomes push against each other.
Oh, so they physically elongate the entire cell.
Exactly. And once the identical chromatids are safely sequestered at opposite ends, telophase kicks in.
It rebuilds the nuclear vaults around them.
Right.
And then cytokinesis actually chops the cell body in half. Animal cells use a ring of actin filaments to pinch the middle, like a drawstring bag, right? The cleavage furrow.
Yep. But plant cells have that rigid cellulose wall, so they can't really pinch.
Right, they have to pave a completely new highway down the middle using vesicles full of cell wall material.
And the end result is two genetically identical daughter cells. Perfect cloning.
Which naturally leads humanity to ask, as our understanding of this perfected mechanism grew, can we hijack this?
Oh, of course we asked.
Right. Like, if mitosis is just a mechanical process, can we force a highly specialized animal cell to act like a fertilized egg?
Which brings us right to Dolly the sheep.
Yeah, the text covers somatic cell nuclear transfer. So, Dr. Ian Wilmot took an udder cell from an adult sheep, a cell that was fully committed to just being an udder cell, and used its nucleus to create a clone.
Right.
But from a cellular perspective, how did he actually wipe the udder cell's memory? Like, how do you tell an adult nucleus to go back to being a blank slate embryo?
That is the real crux of the challenge, because as a cell differentiates during development, certain genes are permanently locked away through epigenetic tagging.
Okay, what does that mean exactly?
It's essentially putting chemical padlocks on the DNA you don't need. An udder cell still has the instructions for building a brain or a heart, but those instructions are locked.
Got it.
So Wilmot's team extracted the nucleus from that udder cell and placed it next to an unfertilized egg cell that had its own nucleus sucked out.
So you basically have a blank egg cell and an adult nucleus just sitting next to it.
Exactly. Then they delivered a very specific pulse of electricity, and this shock did two things.
Okay.
First, it forced the membranes to fuse, dropping the adult nucleus right into the empty egg.
Makes sense.
Second, the electrical pulse actually mimicked the natural jolt of calcium ions that occurs when a sperm enters an egg.
Oh, wow.
Yeah, so this shock, combined with the unique cytoplasm of the unfertilized egg, chemically blasted the padlocks off the adult DNA.
It triggered a massive epigenetic reprogramming.
Exactly. It reset the DNA back to an embryonic state.
So the cell just woke up thinking it had just been fertilized. It started doing mitosis, formed an embryo, and Dolly was born.
Yep. And from there, the textbook jumps to genetic engineering. Not just copying whole genomes, but actually inserting specific genes.
Right. The spider goat is the classic example here. Goats that produce spider silk proteins in their milk.
It's a remarkable application, really. They isolated the gene for spider dragline silk, which is one of the strongest biological materials on Earth.
And they spliced it into the goat's genome right next to the promoter sequence for milk production.
Yes. And a promoter is basically like a switch. It guarantees that the spider silk gene is only turned on in the mammary glands during lactation.
Right, rather than being expressed in the goat's liver or brain or something.
Exactly.
It's just breathtaking technology. But, it leads me back to our aphids.
Oh, the aphids again?
Yeah,
Okay.
because if we look at the sheer biological effort humans put into cloning, and then look at the effortless trillion-fold cloning of the aphid, I really have to push back on the whole concept of sex.
Okay, I see where you're going.
Right. Like, if I have an organism that is perfectly adapted to my current environment, and I can clone myself flawlessly via mitosis, why on earth would I ever engage in sexual reproduction? Mixing my perfect genes with another random individual just seems like a massive downgrade.
That is perhaps the greatest question in evolutionary biology because honestly, you were right. Sexual reproduction is incredibly expensive. We actually call it the twofold cost of sex.
Well,
Yeah. An asexual population grows twice as fast as a sexual one because literally every individual can give birth.
right, you don't need males.
Exactly. Furthermore, think of the metabolic drain of sex. Like a male moose drains massive amounts of calcium from his own skeleton every single year to grow antlers.
Just a fight for a mate.
Just for that. Or a peacock, growing a tail so heavy and conspicuous it actively invites predator attacks.
Not to mention the energy plants spend making nectar purely to bribe bees into carrying their pollen. It's an extortion racket.
It basically is. So why pay the extortion? The answer is the Red Queen hypothesis.
Okay, from Alice in Wonderland.
Well, through the looking glass, specifically, where the Red Queen tells Alice it takes all the running you can do to keep in the same place.
Ah.
In biology, the environment is never static. Most importantly, parasites and pathogens are constantly mutating.
Right, they're always evolving.
So if you are an aphid building a monoculture of 1 trillion perfect clones, you are basically beating 1 trillion identical walks.
Oh, I see.
A parasite only needs to mutate a single key, and it will wipe out your entire trillion-member population in a matter of weeks.
Because none of them have a genetic defense. They put all their evolutionary chips on a single number.
Exactly. Sexual reproduction is really a survival strategy based on chaos. It deliberately scrambles the genetic code every single generation.
So by mixing your genes with someone else's, you ensure your offspring have entirely new combinations of immune defenses.
Right. The parasite's key no longer fits the lock. The cost of sex is high, but the cost of perfect cloning is eventual extinction.
Okay, so the species needs chaos to survive. Let's look at the cellular engine of that chaos, meiosis.
Reduction division.
Right, because if we just fused a normal human sperm and a normal human egg, 46 chromosomes plus 46 chromosomes equals 92.
And the next generation would have 184, the cell would just burst.
We need a reduction division to have the genome, creating haploid cells with just 23 chromosomes.
Which is exactly why meiosis has 2 separate divisions. Meiosis the second and meiosis the second. And if you are studying this for a test, flag meiosis on your mind.
I feel like, yeah.
Because this is where the biological chaos is deliberately engineered.
Let's look at prophase I, because in mitosis, the chromosomes just condense and ignore each other. But in prophase I of meiosis, the homologous chromosomes actively seek each other out.
Right, and let's clarify homologous for the listener.
Good idea. So you have 46 chromosomes, but really it's 23 pairs. One set of 23 from your genetic mother, one set from your genetic father.
Yep.
Chromosome 7 from mom and chromosome 7 from dad are homologous.
Yeah.
They carry the instructions for the same traits, say eye color, even if mom says blue and dad says brown.
Right. And during prophase I, those 2 homologous chromosomes physically zip together along their entire length. This is called synapsis.
Synapsis.
And it's mediated by a protein zipper called the synaptonomal complex. You now have a tetrad 4 chromatids tightly bound together.
And then the enzymes go to work, right?
They do. They deliberately snap the DNA backbones of the non-sister chromatids, mom strand and dad strand, and swap the severed ends.
Crossing over. They literally splice the physical tapes together.
Exactly. And the points where they cross over are actually visible under a microscope. They're called chiasmata.
Oh, wow.
This is genetic recombination at its most fundamental. The chromosome that eventually makes it into the sperm or egg is no longer just grandpa's chromosome or grandma's chromosome.
It is a completely novel patchwork mosaic of the two.
Exactly.
That is the first engine of chaos. The second is metaphase I, because in mitosis, all 46 chromosomes lined up single file to be split.
Right.
But in metaphase I, the homologous pairs line up at the equator. But the way they orient themselves is independent of every other pair.
Independent assortment. Let's say the maternal chromosome for pair number 1 faces the left pole. For pair number 2, maybe the maternal faces left again. But for pair number 3, the paternal faces left.
And because the human haploid number N is 23, the mathematical formula for the number of possible orientations is 2 to the power of 23,
Exactly.
which comes out to over 8.3 million. So just based on how the pairs line up, one human can produce over 8 million genetically distinct gametes.
And that is before you even factor in the infinite variation introduced by crossing over in prophase.
That is just wild.
The mathematical reality is that, short of identical twins, no two humans on Earth will ever share the exact same genetic sequence.
Okay, so anaphase I pulls the homologous pairs apart.
Yeah.
The sister chromatids stay connected.
Right.
We now have two cells, and they are technically haploid. They only have 23 chromosomes each. They just happen to still be duplicated X shapes.
Right.
Then we go immediately into meiosis the second, which is essentially just mitosis for haploid cells.
Yeah.
Line them up, single file, split the centromeres, pull the sister chromatids apart.
And the end result, 4 completely unique haploid gametes.
But the physical realization of those gametes' chemogenesis diverges wildly, depending on whether you are producing sperm or eggs.
Very wildly. Spermatogenesis is optimized for a single variable, motility.
It just needs to move.
Right. The cytoplasm divides equally, resulting in 4 tiny, streamlined sperm cells. They shed almost all their internal organelles, tightly pack their DNA into the head, and use massive numbers of mitochondria to power a flagellum, or a tail.
I mean, they are essentially outboard motors carrying a biological payload.
That's perfectly said.
But the egg cellogenesis plays a completely different game. It doesn't need to move. It needs to build a human.
Exactly. Once fertilized, that single zygote has to immediately undergo rapid mitotic divisions before it even implants in the uterus.
It needs a massive stockpile of energy, ribosomes, and mRNA.
So Genesis uses a brilliant resource hoarding strategy. During both meiotic divisions, the cytoplasm does not split evenly.
The spindle fibers intentionally form off-center.
Yep. One cell takes almost 100% of the cytoplasm and nutrients, becoming the massive ovum. The other nuclei are cast off with practically nothing into tiny packages called polar bodies.
Which quickly undergo apoptosis, right? Programmed cell death.
Exactly. So one primary oocyte yields only one functional, heavily resourced egg.
And before we leave sex entirely, we really have to talk about how biological sex is actually determined. In humans, it's the XXXY system.
Because females are XX, every egg carries an X. Males are XY, so sperm can carry an X or a Y.
And the Y chromosome has a specific gene, the SRY gene, that cascades the development of male anatomy.
Right, but that is just one evolutionary solution. In birds and some reptiles, the system is inverted, the ZW system.
Oh, right.
Yeah, the female is the heterozygous one, ZW, and the male is homozygous, ZZ.
And in social insects like ants and bees, there aren't even sex chromosomes at all. It's a hapodiploid system.
Yep. A queen bee controls the sex of her offspring by just choosing whether or not to release sperm to fertilize the egg as she lays it.
Which is mind-blowing.
Fertilized diploid eggs become female workers. Unfertilized haploid eggs develop parthenogenetically into male drones.
But my absolute favorite is the temperature-dependent sex determination in reptiles. Sea turtles and crocodiles.
It's fascinating.
There is no genetic difference between males and females at conception. It's entirely environmental.
It hinges on the thermodynamics of enzymes. Specifically, an enzyme called aromatase, which converts testosterone into estrogen.
Okay, so how does the temperature affect that?
Well, in many turtles, if the egg is incubated at a warmer temperature, the aromatase enzyme becomes highly active, flooding the developing embryo with estrogen, and it develops as a female.
Wow.
But if the nest is cooler, the enzyme is sluggish, testosterone dominates, and it develops as a male.
Which is terrifying when you think about global warming.
It really is.
A shift of just a few degrees could mean a sea turtle population only produces females, driving the local population to extinction.
It is a profound vulnerability, and it highlights how delicate these biological cascades really are, which actually brings us to the inevitable consequence of all this complexity: mistakes.
Right, we've talked about the perfection of mitosis and the choreographed chaos of meiosis. But what happens when the machinery physically breaks down?
Yeah, what happens if, during anaphase, the motor proteins on the kinetochore fail, or the cohesion holding the chromosomes together just doesn't dissolve?
We call that non-disjunction, right?
Yeah.
It is a mechanical failure of separation.
Exactly. If homologous chromosomes fail to separate in meiosis first, or if sister chromatids fail to separate in meiosis second, the resulting gametes will have an abnormal number of chromosomes.
A condition called aneuploidy.
Right.
So instead of a perfect 23, a gamete ends up with 22 or 24.
Yeah.
And if that gamete is involved in fertilization,
The resulting zygote will have 45 or 47 chromosomes. Missing a chromosome is a monosomy. Having an extra is a trisomy.
and because genes rely on incredibly precise protein dosage, Having an entire extra chromosome's worth of active genes throws the cellular economy into total chaos.
Which is why most autosomal aneuploidies are lethal early in embryonic development.
But a few are survivable. Trisomy 21 is the most common, resulting in Down syndrome.
Right. Chromosome 21 is the smallest human autosome, and it contains relatively few genes, which is why a trisomy here is compatible with life.
It occurs when an individual inherits 3 copies of chromosome 21.
And we know that the incidence of non-disjunction increases significantly with the maternal age of the oocyte.
Right, because as an egg cell pauses in prophase for decades, the molecular machinery, like those cohesin proteins holding the tetraps together, can naturally degrade.
Exactly.
Now, the textbook mentions prenatal testing to detect this, things like amniocentesis or CVS. How are they physically checking the chromosomes of a fetus?
They create a karyotype. Using ultrasound guidance, doctors extract a sample of fetal cells, either from the amniotic fluid or the chorionic villi of the placenta.
Okay.
They culture those cells in a lab and then hit them with a chemical that halts mitosis right in metaphase when the chromosomes are most densely coiled and visible.
Oh, that's clever.
Yeah. They stain them, photograph them, and digitally arrange them into their 23 homologous pairs. A simple visual inspection reveals if there is a trisomy or monosomy.
And non-disjunction can happen with sex chromosomes too, right?
Definitely.
Like Turner syndrome is XXY a female with only 1 X chromosome. Klinefelter syndrome is XXY a male with an extra X. The cellular machinery just misfires.
But sometimes the machinery works perfectly, but the gametes just can't physically reach each other, which is where assisted reproductive technologies come in.
Right, because about 10% of couples face fertility challenges. In vitro fertilization, or IVF, is the primary intervention.
It involves hormonal stimulation to mature multiple eggs, extracting them surgically, and mixing them with sperm in a highly controlled laboratory environment to ensure fertilization.
But what if the sperm's flagellum is defective? What if it literally cannot swim?
Well, natural selection will usually end the line right there.
But we have ICSI, intracytoplasmic sperm injection.
It is a total triumph of micromanipulation. A technician uses a microscopic glass pipette to hold the egg cell perfectly still, and another incredibly fine hollow needle to pierce the zona pellucida, the egg's tough outer shell. And they inject a single, specifically chosen sperm directly into the cytoplasm. It completely bypasses the need for sperm motility or the natural enzymatic breakdown of the egg's defenses.
Okay, I want to circle back to something we touched on earlier because it breaks one of the biggest rules of genetics.
Let's do it.
We keep talking about the nucleus as the absolute vault of DNA, but it isn't. The mitochondria powering our cells have their own DNA.
Cytoplasmic inheritance. And that goes back to the endosymbiotic theory.
Right. Billions of years ago, the ancestor of our eukaryotic cells was this large, sluggish microbe, and it swallowed a smaller, highly efficient oxygen-breathing bacteria.
But instead of digesting it, they formed a symbiotic relationship.
Exactly. That swallowed bacteria eventually evolved into the mitochondrion, but it never gave up its own circular chromosome.
And because of how oogenesis works, because the egg hoards all the cytoplasm and the sperm strips it all away, this creates a completely different inheritance pattern.
Yes. When the sperm penetrates the egg, it delivers its nuclear payload, but the sperm's mitochondria are actively destroyed by the egg's defense mechanisms.
Therefore, 100% of the zygote's mitochondria come from the mother.
Your mitochondrial DNA is a direct, uncloned copy of your mother's and her mother's, stretching back hundreds of thousands of years.
It completely ignores Mendelian rules.
Mendelian rules, which is the perfect transition to the next phase of our review. We know chromosomes move, we know they trade pieces, but how does this microscopic shuffling actually result in you having brown eyes and me having blue?
To answer that, we have to travel back to an Austrian monastery in the 1800s and look at the work of Gregor Mendel.
What is truly staggering about Mendel is that he deduced the precise mathematical laws of heredity without ever looking through a powerful microscope.
He didn't know what DNA was.
He didn't know chromosomes existed. He treated heredity as a mathematical black box.
He used pea plants. And he didn't just casually observe them, he rigorously controlled their reproduction.
He started by breeding plants until he had true breeding lines. Like if a plant had purple flowers, he bred it with itself for generations until he was absolutely certain it would only ever produce purple flowers.
He established his baseline. And we need to lock down the vocabulary he established here because it is literally the grammar of genetics.
Right. So he hypothesized that traits are controlled by discrete units of inheritance, which we now call genes.
And the alternative versions of a gene are alleles.
So the gene determines flower color. The alleles are the specific instructions, purple or white.
Exactly. And because pea plants are diploid, they have two alleles for every gene. If the alleles are identical, say, to purple alleles, the plant is homozygous.
And if they are different, one purple, one white, it is heterozygous.
Right. And the specific combination of those letters, like big P, little p, is the genotype. What the plant actually looks like, the purple flower, is the phenotype.
Okay, so Mendel initiates his first major experiment, the monohybrid cross.
Right. He takes a true breeding tall plant and forcefully cross-pollinates it with a true breeding dwarf plant. This is the parent, or P generation.
And at the time, the prevailing scientific dogma was the blending hypothesis. Everyone basically assumed the offspring would be medium-sized.
Like mixing paint.
Right, but they weren't. The entire first generation, the F1 generation, grew to be strictly tall. The dwarf trait vanished completely.
Which completely destroyed the blending hypothesis. Mendel concluded that the tall allele was dominant over the dwarf allele.
If the tall allele was present at all, it dictated the phenotype.
And the dwarf allele was recessive. It was chemically present in the plant's genotype, but it was completely masked.
But I really want to pause here and explain the mechanism of dominance, because I feel like textbooks often make it sound like the dominant allele is actively bullying the recessive one.
Oh, yeah, they do?
That's not what's happening at the biochemical level, is it?
No, not at all. It's usually just about enzyme production. A dominant allele typically codes for a functional protein or enzyme. The recessive allele is often just a broken or mutated version of that same gene that produces a non-functional enzyme, or maybe no enzyme at all.
Right. So in the case of plant height, the dominant tall allele produces an enzyme that synthesizes a growth hormone called gibberellin.
Exactly. The recessive dwarf allele has a typo in its DNA, so it produces a broken enzyme.
If a plant is heterozygous, it has one good allele making gibberellin and one broken one doing nothing.
But that one good allele produces enough hormone to make the plant grow tall.
The functional allele just compensates for the broken one. That makes perfect sense. Okay, so Mendel has these tall F1 heterozygotes. He lets them self-fertilize to create the F2 generation.
And suddenly, the dwarf trait comes back.
Yes, out of thousands of plants, he observes a strict mathematical ratio. 3 tall plants for every 1 dwarf plant.
From this 3.1 ratio, Mendel derived his first law, the law of segregation.
He stated that an organism's 2 alleles for a trait must separate cleanly during the formation of gametes. Each pollen grain or egg cell receives only 1 allele.
And this is where Mendel's genius really shines. He deduced the exact mechanism of anaphase elite of meiosis homologous chromosomes, separating decades before anyone actually saw a cell divide.
It's incredible, and the Punnett square is just a visual representation of this segregation. You put the 2 possible alleles from one parent on the top and the 2 from the other parent on the side, and multiply them to see the 4 possible zygotes.
The Punnett square also highlights a practical problem breeders face, though, which led to the test cross.
Right. Because if you see a tall pea plant, you know its phenotype, but you don't know its genotype. It could be homozygous dominant, TT, or heterozygous TT.
So how do you find out without sequencing its DNA? You interrogate it with a test cross, you breed that mystery tall plant with a dwarf plant.
Why a dwarf? Because a dwarf plant has a known genotype. It must be homozygous recessive, little t.
Exactly. You look at the offspring. If the mystery plant was TT, it will only give dominant T alleles, and 100% of the offspring will be tall.
But if the mystery plant was hiding a recessive allele T, then half the time it will pass on the little t.
Right. Combined with the dwarf parent's little t, you will see a 1.1 ratio of tall to dwarf offspring. The hidden genotype is basically revealed by the phenotype of the next generation.
Okay, so tracking one trait is pretty straightforward. But Mendel wanted to know if traits were physically tethered together. Does being a round seed mean you automatically have to be a yellow seed?
This leads to the dihybrid cross, tracking 2 traits simultaneously.
So he took a plant that was true breeding for round yellow seeds, dominant for both, and crossed it with a plant true breeding for wrinkled green seeds, recessive for both.
The F1 generation was completely heterozygous for both genes, round and yellow. But when he crossed those F1 heterozygotes together,
he didn't just get round yellow and wrinkled green. He got completely new recombinations. He found a massive 16-part ratio. 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green.
The fact that the traits decoupled and mixed in these new combinations led to his second law, the law of independent assortment.
The alleles for seed shapes separate into gametes entirely independently of the alleles for seed color.
Which, again, perfectly maps to metaphase I of meiosis. The chromosome carrying the shape gene lines up completely independently of the chromosome carrying the color gene.
But if you are staring at a dihybrid or trihybrid cross problem on your test, listener, please do not spend 10 minutes drawing a massive 16 or 64 box Punnett square. Use the product law of probabilities.
Oh, yes. It is the most powerful shortcut in genetics. The product law states that the probability of two independent events occurring together is the product of their individual probabilities.
Right. Think of flipping two coins. The chance of coin A landing on heads is 1/2. The chance of coin B landing on heads is 1/2. They don't affect each other. So the chance of flipping two heads at the same time is 1/2 times 1/2, which is 1/4.
You just multiply the fractions, and you apply this directly to the genetic cross.
If you cross two heterozygous parents, RRY times RRY, and the test asks for the probability of getting a completely homozygous recessive offspring Y, you break it down into two simple monohybrid crosses.
What is the chance of getting R times RY? It's 1/4. What is the chance of getting A from Y times YY? Also 1/4.
Multiply them together. 1/4 times 1/4 is 1/16. You arrive at the exact same answer as the giant Punnett square in 10 seconds.
Math is the ultimate cheat code.
It really is. But here's the catch. Mendel was incredibly lucky. He chose traits in peas that were strictly dominant or recessive and just happened to be located on different chromosomes. But biology is rarely that neat.
True. In the real world, the rules break down. This brings us to complex inheritance. What happens when an allele isn't a dictator?
Let's start with incomplete dominance. This occurs when a heterozygote displays an intermediate phenotype that is a blend of the two homozygous phenotypes.
The classic textbook example is the snapdragon flower. Cross a red snapdragon with a white snapdragon, and the F1 generation is entirely pink.
But again, let's look at the mechanism. Why is it pink? It's not because the red and white alleles are literally mixing together like liquids. It goes back to enzyme dosage.
Right. The red allele codes for an enzyme that produces red anthocyanin pigment. The white allele has a mutation and produces a broken enzyme. So, no pigment.
If you are homozygous red, you have two pigment factories running at full capacity, so the flower is deep red.
And if you are heterozygous, you only have one functioning factory. It produces pigment, but only half the normal amount.
That lower concentration of red pigment in the petals visually appears to us as pink. It is just a dosage effect.
Exactly. Contrast that with codominance, where both alleles are fully dominant and fully expressed at the exact same time. There is no blending.
The textbook uses roan cattle. You cross a red bull with a white cow, and the calf isn't pink, it's roan.
If you look closely at a roan calf's coat, you won't see pink hairs. You will see individual, fully red hairs intermingled with individual, fully white hairs.
Both alleles are being fully transcribed and translated, just in different skin cells.
And the most critical human example of codominance is our ABO blood type system, which also introduces the concept of multiple alleles.
Right, because Mendel's peas only had two options, tall or short. But human blood type has three possible alleles floating around in the population, IA, IB, and lowercase I.
These genes code for an enzyme called a glycosyltransferase, which attaches specific sugar molecules antigens to the outside of your red blood cells.
The IA allele attaches A-type sugars. The IB allele attaches slightly different B-type sugars. And the recessive I allele is a broken enzyme that attaches nothing at all.
So if you inherit the IA allele from mom and the IB allele from dad, your cells build both enzymes. You attach both A and B sugars equally all over your red blood cells. You are blood type AB.
Complete codominance.
But if you inherit two recessive I alleles, your red blood cells have a smooth surface with no A or B antigens. You are type O.
And this has profound medical implications. Your immune system is trained to attack any foreign antigens. If you have type A blood and someone transfuses type B blood into you, your antibodies will attack those B sugars, causing the blood to clot fatally.
But type O blood has no antigens on its surface. It wears an invisibility cloak.
Which is why type O negative is the universal donor. The recipient's immune system has nothing to attack.
Okay, what about traits that aren't discrete categories? Humans aren't just 5 feet tall or 6 feet tall. We have continuous variation.
That is because traits like height, skin pigmentation, and eye color are polygenic. They are controlled by the additive effect of multiple genes, often spread across different chromosomes.
Skin color in humans is a prime example. Melanin production isn't a single on-off switch. It is controlled by dozens of genes.
So every dominant allele you inherit for one of those melanin-producing genes acts like a tiny scoop of pigment added to a bucket.
If you inherit mostly dominant alleles across all those different genes, you produce a lot of melanin and have very dark skin.
If you inherit mostly recessive alleles, you produce very little melanin. It creates a smooth bell curve of phenotypes across the population.
And because humans have these incredibly complex polygenic traits, and because we produce very few offspring over long generation times, we obviously can't study human genetics the way Mendel studied peas.
Right, we have to work backward through history using pedigrees.
A pedigree is basically a genetic family tree. It's detective work. On a test, you'll see squares representing males and circles representing females.
Shaded shakes mean the individual expresses the trait. By analyzing how a disease travels through the generations, you can deduce its mode of inheritance.
For instance, if a disease is autosomal recessive, like cystic fibrosis, you will often see it skip generations.
CF is caused by a mutation in the CFTR gene, which codes for a channel protein that pumps chloride ions across cell membranes.
When it's broken, water doesn't follow the ions, resulting in a buildup of thick, sticky mucus in the lungs and pancreas.
Because it's recessive, a person must inherit two broken copies. So you will often see two unshaded, healthy parents, who are secret heterozygous carriers, suddenly have a shaded child.
The textbook also highlights PKU phenylketonuria, another autosomal recessive disorder where the body fails to produce the enzyme needed to break down the amino acid phenylalanine.
If it builds up, it causes severe brain damage. But here's the incredible part. Because we understand the genetics, we can intervene environmentally.
Yes, every newborn is screened for PKU via a heel prick blood test. If they have it, they are simply put on a strict diet free of phenylalanine and they develop completely normally.
The genotype remains broken, but we manipulate the environment to fix the phenotype.
Exactly. But the most famous and historically devastating patterns in pedigrees belong to sex-linked traits. Remember that the X chromosome is massive, carrying thousands of crucial genes.
The Y chromosome is tiny, essentially just carrying the switch for male development.
This creates a massive structural vulnerability for biological males. We are homozygous.
For any gene on the X chromosome, a male only gets one copy. Whatever allele is on that single X chromosome is exactly what gets expressed. There is no backup copy to mask a mutation.
Which is why X-linked recessive disorders like red-green colorblindness or Duchenne muscular dystrophy overwhelmingly strike males.
A female would have to inherit two mutated X chromosomes, one from her mother and one from her father, which is statistically rare.
But a male only needs to inherit one mutated X from his mother. The father's Y chromosome offers zero protection.
And the most dramatic historical example of this is Queen Victoria of England and hemophilia.
Hemophilia is an X-linked recessive disorder where a mutation breaks the genes that produce blood clotting factors, specifically factor VIII.
A minor cut or internal bruise can cause fatal bleeding.
Queen Victoria was a heterozygous carrier. She had one mutated X chromosome and one normal one.
The normal one produced enough clotting factor for her to live a healthy life, but she passed that mutated X chromosome down to several of her children.
And because the royal houses of Europe used marriage as a political alliance, effectively creating a highly isolated breeding population, that single mutated allele spread like wildfire.
It did. It devastated the Spanish and Russian royal families. Tsarevich Alexei of Russia, the heir to the Romanov throne, inherited the mutated X chromosome from his mother, Alexandra, who was Victoria's granddaughter.
His severe hemophilia led the family to rely on the mystic Rasputin for healing, which profoundly destabilized the Russian monarchy right before the revolution.
The entire political landscape of the 20th century was arguably reshaped by a single missing sequence of DNA on one X chromosome.
A microscopic typo collapsing an empire.
Which brings us, finally, to the fundamental question. Section 6, the atomic level. Throughout this entire discussion, we've talked about genes and chromosomes and alleles as if they are abstract concepts.
But what physically are they? What is the actual chemical structure that allows for this flawless copying and this immense storage of data?
The hunt for the physical identity of the gene is one of the greatest detective stories in modern science. In the late 19th century, scientists knew chromosomes existed, but they assumed the genetic material had to be proteins.
Proteins are complex. They are built from 20 different amino acids. They seem like the only molecules sophisticated enough to store the instructions for a human being.
DNA, by contrast, seemed incredibly boring. It was first isolated by a Swiss chemist, Friedrich Miescher, in 1869.
He collected pest-soaked bandages from a local hospital, broke open the white blood cells, and extracted a strange phosphorus-rich substance from the nucleus. He called it nuclein.
But because DNA only has 4 building blocks, scientists thought it was too simple to be the genetic code. They thought it was just a structural scaffold for the proteins.
It took brilliant experimental design to prove otherwise. Let's look at Joachim Hammerling in the 1930s. He worked with an organism called a Sacabularia, a single-celled marine algae.
But it is a gargantuan single cell, up to 5 centimeters long, with a distinct cap, a stalk, and a foot containing the nucleus.
Hammerling basically did microscopic Frankenstein experiments. He amputated the caps, the algae grew them back.
But if he amputated the foot, the part with the nucleus, the cell couldn't regenerate. It died.
He even grafted the stalk of one species onto the foot of another, and the newly regenerated cap always matched the species of the foot.
He proved beyond a doubt that the nucleus was the absolute command center.
But the nucleus contains both DNA and protein. The definitive proof came in 1952 with Alfred Hershey and Martha Chase.
They used bacteriophages viruses that exclusively infect bacteria. A virus is the ultimate stripped-down biological machine. It is literally just a hollow protein shell wrapped around a core of DNA.
When a virus attacks a bacteria, it injects its genetic material inside to hijack the cell, while the empty shell stays on the outside.
So Hershey and Chase set up a brilliant chemical trap using radioactive isotopes. They knew proteins contain sulfur, but DNA does not.
And they knew DNA contains phosphorus, but proteins do not.
Exactly. So they grew one batch of viruses in a radioactive sulfur soup, permanently tagging the viral protein coats. They grew a second batch in radioactive phosphorus, permanently tagging the viral DNA.
They let both batches infect bacteria and then put the mixture into a standard kitchen blender.
Literally a wearing blender. They turned it on just fast enough to shear the empty viral shells off the outside of the bacteria.
Then they spun the samples in a centrifuge. The heavy bacteria sank to the bottom into a pellet. The light viral shells stayed suspended in the liquid.
What they found changed biology forever. In the sulfur batch, the radioactivity stayed in the liquid. The protein never entered the cell.
But in the phosphorus batch, the radioactive DNA was found inside the bacterial pellet. The DNA had physically entered the cell and was actively producing new viruses.
It was the smoking gun. DNA was the hereditary material. The debate was over.
But that just shifted the question. How does this simple, boring molecule store information?
We knew its chemical composition thanks to Phoebus Levine in the 1920s. He discovered the nucleotide, the Lego brick of DNA.
A single nucleotide consists of 3 parts. A pentose sugar, specifically deoxyribose, a 5-carbon ring.
Attached to one side of the sugar is a negatively charged phosphate group.
Attached to the other side is a nitrogenous base. And there are 4 distinct bases, adenine, or A, and guanine, or G, which are large double-ring structures called purines.
And thymine, T, and cytosine C, which are smaller, single-ring structures called pyrimidines.
And then Erwin Chargaff drops a massive clue regarding how these bases interact. He analyzed the DNA of dozens of species, from bacteria to humans.
And he found a bizarre mathematical constant. The amount of adenine in a sample always perfectly matched the amount of thymine. A equals T.
And the amount of cytosine, perfectly matched guanine, C equals G.
Chargaff's rules. They strongly implied that A was physically tethered to T and C was tethered to G.
But nobody knew what the molecule actually looked like in 3-dimensional space. That sparked the great race of the early 1950s.
James Watson and Francis Crick at Cambridge were trying to build physical, theoretical models using wire and metal plates, but they were failing because they lacked empirical measurements.
Which brings us to Rosalind Franklin at King's College, and I want to dig into exactly what she was doing, because calling it taking a picture of DNA is an insult to the physics involved.
She was performing X-ray crystallography.
It is an agonizingly precise science. She had to extract DNA, draw it into incredibly fine, highly ordered glass-like fibers, and shoot a concentrated beam of X-rays through it.
As the X-rays hit the atoms in the DNA, they scattered diffracted onto a piece of photographic film behind the sample.
The resulting image isn't a literal photograph of the molecule. It is a mathematical shadow, a pattern of spots.
Her most famous image, photo 51, took 100 hours of exposure time to capture. It showed a distinct X pattern made of dark spots.
And to a crystallographer, an X pattern is the undeniable mathematical signature of a helix. The distance between the spots even revealed the exact width of the helix and the distance between each turn.
But here is the historical tragedy. Maurice Wilkins, a colleague who had a strained relationship with Franklin, showed Photo 51 to James Watson without her knowledge or explicit permission.
When Watson, who understood the math of helices, saw the image, the final pieces clicked into place. Watson and Crick used her precise atomic measurements to build their model.
They published their legendary paper in 1953 proposing the double helix.
And they won the Nobel Prize. Franklin did not. She passed away tragically from ovarian cancer at age 37, heavily exacerbated by her constant exposure to X-ray radiation.
The Nobel Committee does not award prizes posthumously. Watson and Crick were brilliant synthesizers, but it was Franklin's grueling, dangerous experimental data that provided the key.
It's a stark reminder that the history of science isn't just a march of objective facts. It is driven by human ambition, collaboration, and sometimes intense ethical gray areas.
But the model they arrived at, regardless of the messy history, is the most elegant structure in the universe: the double helix.
Imagine a twisted ladder. The two vertical side rails, the backbone, are made of alternating sugar and phosphate molecules.
The bonds holding them together, phosphodiester bonds, are incredibly strong covalent bonds. The backbone is the protective structural armor of the molecule.
But the rails are anti-parallel. One side runs right side up and the other runs upside down, chemically speaking,
Correct.
which is necessary for the rungs of the ladder to fit together.
Exactly. The rungs of the ladder are the nitrogenous bases reaching inward from the sugar backbone. This is where Chargaff's rules become physical reality.
A large purine always pairs with a small pyrimidine, keeping the width of the ladder perfectly uniform. Adenine reaches across and forms two hydrogen bonds with thymine.
Cytosine reaches across and forms 3 hydrogen bonds with guanine.
Hydrogen bonds are the key here. They are weak forces of attraction. They act like molecular Velcro.
They are strong enough to hold the two halves of the DNA ladder together when it's just sitting there, but weak enough that enzymes can easily rip the ladder down the middle when it needs to be copied.
And that complementary base pairing, the fact that A only bonds with T and C only bonds with G, is the ultimate secret of life. It provides the mechanism for perfect replication, semi-conservative replication.
When a cell undergoes mitosis, which is exactly where we started this entire review, an enzyme called helicase unzips the DNA ladder down the middle, breaking those weak hydrogen bonds.
You are left with two single separated strands, exposing the sequence of bases. And because of the pairing rules, each half serves as an absolute foolproof template for a new strand.
If the exposed strand reads ACTG, the cellular machinery simply locks the matching puzzle pieces into place. TGAC.
From one double helix, you flawlessly synthesize two identical double helices. The instruction manual physically copies itself.
And the sequence of those rungs, the millions of A's, T's, C's, and G's running down the length of the molecule, that sequence is the actual code.
It is the syntax of the gene, a sequence that tells a ribosome exactly how to build the gibberellin enzyme to make a pea plant tall, or the hemoglobin protein to clot royal blood, or the maintenance protein to keep a stolen chloroplast functioning inside a sea slug.
It all comes back to the sequence, and that brings us full circle. From the perfect trillion-fold cloning of an aphid to the deliberate chaotic scrambling of chromosomes in meiosis.
We've seen how Mendel found the mathematical probabilities hidden in that chaos, how blood types and genetic disorders break those simple rules, and how a molecule that looks like a twisted microscopic ladder manages to dictate the shape of every living thing on this planet.
So when you walk into your unit test, do not view these concepts as isolated chapters. You are tracing the flow of biological information.
You are tracking how life stores its data, how it perfectly replicates it to survive today, and how it deliberately mutates it to survive tomorrow.
You know the mechanisms now. You know exactly why a spindle fiber tugs on a kinetochore and how the dosage of an enzyme turns a flower pink. Trust your understanding of the systems.
The vocabulary will naturally fall into place because you actually understand the architecture underneath it.
We will leave you with this final thought. Over the last few decades, humanity has done something unprecedented. We have sequenced the human genome. We know the exact order of the 3 billion A's, T's, C's, and G's that make us up.
But knowing the alphabet is not the same as understanding the poetry.
As you look at your test, and as you watch revolutionary tools like CRISPR gene editing advance in the coming years, remember this. We have spent the last century figuring out how to read the code of life. Your generation is going to be the one that has to figure out the profound consequences of rewriting it.